Thank you for that very kind introduction. And I'm frankly slightly intimidated following up Lucas's fantastic talk, but I hope I can keep your interest over the next about half an hour. And in keeping with today's sort of theme of applications in biology with the use of optogenetics, I thought I would present a talk on combining fast 3D confocal imaging and photomanipulation on our flagship microscope, which is the Andor Dragonfly600. A brief overview of what the next 20 minutes together are going to look like. I'll briefly introduce you to who we are and what we do. I will introduce you to our spinning disk microscopes, and then I will keep most of this talk focused on photostimulation using a spinning disk microscope and how we might go on and do that. And since I'm an application scientist, I'm most interested in the applications themselves, right? So what can you do with these devices? So I will sort of round off the talk talking about a few different case studies where these devices have been used by people and me to do their science. So I'll start off by introducing Andor to you guys. We're part of the Oxford Instruments group. We're a small company. Our manufacturing unit is based in Belfast in Northern Ireland. And we make a variety of tools and devices that allow you to do a lot of scientific imaging, spectroscopy, and microscopy as well. Since we were established in 1989, we've continuously driven innovation, but primarily keeping our focus on microscopy systems, we specialize on spinning disk confocal microscopes. We released our first series of spinning disk systems called Revolution series in 2005. And at the time, we were partnering with Yokogawa and using their spinning disk design. But since then, we've refined that design and patented our own spinning disk now. And we use that in our Dragonfly series, which was launched in 2016. Since then, we've made a benchtop version of a spinning disk system, which I will introduce you to in a bit. And then that was followed on by an improvement in the Dragonfly series to make a comprehensive imaging solution, or as comprehensive as we could get. And we will talk about these two systems in a bit more detail. And as of earlier this year, we have been invited to join the industry board of Euro-BioImaging, and we're very proud and collaborators of this community. So thank you for having us. As I just said, our spinning disk systems come in two flavors. So there is the benchtop version, which is BC43, and there is the highly configurable platform called the Dragonfly. Now, of course, this audience does not need an introduction to the technology itself. So it just suffices to say that they're really well suited to do your fast 2D and 3D acquisitions, and particularly for live cell imaging experiments. Our benchtop microscope, which is BC43, is essentially the size of a lab centrifuge, as you can see on the image here on the right. You do not need a darkroom or any vibration isolation table. So it makes it extremely compatible to do your experiments and then take it right through to microscopy. And this is really crucial when you're doing live cell imaging experiments, right? You don't want that time delay between sample prep and then imaging. So this system is quite robust and allows you to do just that. It is a 4-laser system, so it's quite nice to image your blue, green, red, and far red dyes, which are the most commonly used dyes when you multiplex. And while it's predominantly a spinning disk system, you can also use it in wide field or bright field modalities to image your samples. So in just this little box here, you get quite a versatile imaging solution that allows you to look at cellular and subcellular structures over time, but you can also be imaging really large tissues like this mouse embryonic heart section on the right. And this particular example is one where the image was stitched together using, after acquiring multiple tiles, and you're able to acquire this sort of, or attain this sort of perfect stitching because of how we deliver the illumination. And so this is another patented technology at Andor. We call it Borealis illumination, and we aim to deliver an even flat field illumination to your samples. So when you image these large samples, then you don't, aesthetically, well, doesn't come out looking like a quilt, which is important. But also, if you were wanting to do quantitative biology, then it's important that your signal strength does not sort of dip off at the edges of your field of view. So I hope you're impressed by this benchtop solution here. I'll now very quickly introduce you to our Dragonfly series, which is an extremely flexible and multimodal microscopy platform. Again, predominantly a spinning disk system. You can have up to 2 cameras, and you can choose between EMCCD or SCMOS cameras, depending on the samples, on your applications. And you can achieve true simultaneous dual camera acquisition with scan speeds as high as 400 frames per second. It comes with a high power laser engine that gives you a much better penetration into deeper tissues if you're wanting to image organoids or large tissue sections, etc. And you can have up to 10 laser lines, so you can image a variety of dyes going all the way from your UV to infrared part of the spectrum. Now, along with that, you have the choice to add the optics to do turf. You also have the choice to combine that with astigmatic encoding and do super resolution imaging for SMLM type applications. So it's an extremely configurable model that allows you to cater to a wide range of applications, really. You can also improve your resolution using a camera-based technique called SurfStream, which does not require any extra addition of hardware. So it's extremely cost-effective in its ability to provide you a super resolution image. Now, what all of this really means in context of your science is that on the same system, you could be imaging really large tissues like this chick embryo head, which is 1.2 millimeters deep, has been imaged in 3 colors, over 220 planes in Z, and stitched together using a 7x5 montage. And all of this was done in about 2 hours. So it really sort of improves your acquisition speeds. But on the same system, you can also be looking at really fine subcellular details, right? You can be looking at a turf time lapse, like here on the right, and you could be looking at beta integrin dynamics. So this really saves you the trouble of having to learn multiple softwares, having to move to different systems to acquire various types of images for various applications and so on. We're, of course, also aware that a lot of biology is lately done using photomanipulation experiments. And like Lucas pointed out, it's becoming more and more common to use optogenetics, uncaging, photoconversion, or even ablations to perturb your sample optically within a defined region of interest, and then use this perturbation to elucidate structural or functional properties of the specimen. Now, the bottom line of wanting to do any and all of these types of techniques is the need to target light to a region of interest, right? So now if we take a step back and just look at the workings of a spinning disk microscope, we know that here we're working with multiple pinholes versus the single pinhole that a laser scanning microscope has. And what this means in context of image formation is that in a laser scanner, you typically have a galvanometer mirror that guides this laser across your specimen, and then your image is built pixel by pixel. In contrast, in a spinning disk system, light is dispersed through a spinning disk, and your sample is illuminated at multiple points simultaneously, and an image is formed. And this image is usually captured by a camera. What this inherently means is that in a laser scanner, you have the ability to park your laser at a desired region of interest. So most people will prefer a laser scanner when they do a photostimulation experiment. And this works very well for a wide range of applications. But if you were interested in combining that with fast acquisitions, maybe because your biology moves quite rapidly and you need that type of scan speed to capture the events post your photostimulation, then you want to try and combine photostimulation with a spinning disk system, right? And at Andor, we've got 2 different technologies that allow you to arrive at that combination. And these are based on 2 different techniques altogether. So on the one hand, we have MicroPoint 4, which was recently launched last year, And what it is a galvo-guided laser scanning based illumination system. So much like a point scanner, essentially. There is a galvo mirror that guides the laser, and you're thus able to park the laser at a desired point or region of interest on your sample. On the other hand, you have Mosaic, which is a digital mirror device-based illumination system. I'll talk about both of these in a bit more detail, but what I'd like to emphasize here is that both of these tools or devices utilize the back epi-illumination port on your inverted microscope. What that means is that our confocal unit, which is the Dragonfly, or any confocal unit, your spinning disk unit you're working with, is now free to use the side port, which is where it usually sits. So we're not disturbing the spinning disk configuration at all. We're just adding on a device that uses the back port. And thus you're able to combine these two technologies and do your photostimulation experiments on a spinning disk system. Let's first talk about the Micropoint 4. As I mentioned, what it has is fast scanning galvos that allow for precise beam steering, and you can park the laser at a point or region of interest. Now, for a laser source, you could either use a nitrogen pumped dye laser, which is a pulsed laser delivery system. This comes with 24 dye cells, so it allows you the flexibility to do your photostimulation anywhere from 365 nanometers to 656 nanometers. Or you could be using a continuous wave laser engine, which is the same sort of laser engine that you would use for imaging. So if you were doing this on the Andor Dragonfly, then you could use the high-powered laser engine that I just mentioned when talking about the microscope. And a second port on this laser engine would be used to give you an input into the Micropoint 4. So what can you do with this tool, right? And that's what I'm most interested and excited about. So I'll show you some examples where I've used the Micropoint 4, and others have used it too to do their work. The first example I'm going to show you now is of a targeted ablation of a microtubule bundle in a human fibroblast cell. So if you keep your eyes peeled at where the arrow is pointing, you're now able to see a very precise ablation of this particular microtubule bundle without disrupting the rest of the structure. And I want to show this example just to sort of illustrate what kind of precision you can achieve with this device. And a lot of axotomy experiments are done almost uniquely with Micropoint 4 lately. The second example I'm going to show you today is one of wound healing dynamics. So this data was submitted to us at the Andor Insight Awards, about which I'll talk to you more later. Thank you to Mitsutoshi Nakamura for sending us this data. What they're doing here is they're looking at a Drosophila cell. They've used the Micropoint 4 to make a wound, and then they're looking at the effectant dynamics. And you can see this beautiful contractile ring that's formed at the periphery of the wound and helps its eventual closure. So this is a great example of where you want to combine your wound, your wounding, and then you want to follow that up with really fast time-lapse acquisition to capture this sort of dynamics. And the last case study I'd like to present here is something that came out of a lovely workshop that I did with Bénédicte Charrier at the microscopy school in France called Mifobio. This takes place every 2 years, and it's an excellent environment to really just immerse yourself in all things microscopy. So Bénédicte is based in Lyon, and what she does is she looks at the division and orientation dynamics of brown algae called Fascillaria. And these are fascinating organisms because they determine their morphology one cell division at a time. So what that means is that every cell has a say in the division and the orientation of the cell adjacent to that. And her preferred method of studying these orientation dynamics is using microablation. So here we're using Micropoint 4 to make a fine incision in one cell alone. And you can see in the corresponding fluorescent image that all the chloroplasts from that cell have been released into the environment. But we've done this without compromising the integrity of the rest of the filament. And that's quite important because what she essentially wants to do is put this filament back in culture and then observe the division dynamics over many days or hours. So I hope I've given you a sort of a flavor of what you can do with combining micropoint 4 and a spinning disk confocal microscope. And I'd like to do the same with the other device I mentioned, which is Mosaic. So just as a brief reminder, Mosaic is a DMD device. So these are digital mirror devices. At its heart is an 800 by 600 array of mirrors. And these are tiltable mirrors. So what happens is that when you send a short pulse or a short voltage, these mirrors tilt and thus guide the light to your sample. So you could draw these sort of regions of interest on your sample, and then that will send a short voltage to all the mirrors within that region, and thus you achieve targeted illumination within your region of interest. It's important to note that with the Mosaic, you could be doing this simultaneously across various regions of interest. I'm going to do the same for the Mosaic as I did for the micropoint. Just going to show you a couple of examples where the Mosaic has been used for really fascinating work. So the first example here is also something that came out of Andor Insight Award submission from Weiwei Zhang at Purdue University. And here we're looking at an Arabidopsis leaf epidermis. And what they've done here is to use the Mosaic to target light to this small region of interest here in the center. They've then used a spinning disk. In this case, actually, they had a revolution, I believe. And they've used that to capture this beautiful radial calcium wave that spreads outwards from this point of ablation. And this, I think, again, just really shows you the strength of fast acquisitions post photostimulation, because you need that sort of frame rate to capture the event seamlessly. And I'll show you one last example where I used the Mosaic recently. And this was for a project with Milka Saris at the anatomy department in Cambridge. And she wants to look at neutrophil migration during inflammation. So she has designed a zebrafish line where she's able to photoconvert neutrophils around the site of inflammation. And post-conversion, she would like to monitor the migration of neutrophils throughout the organism. And what that means is that we need to be able to image the animal in 3D. So we're talking Z stacks. And we need to be able to tile images so as to capture the entire animal. And we want to do a time-lapse acquisition. So that's what I'm going to show you now. So you can see the converted cells here in red. And then I've used our analysis software, Imaris, to segment out a control population of non-converted cells and these converted cells here in purple. And now you're able to compare their displacement lengths. In this case, of course, we're looking at an unchallenged fish. So the converted cells are not moving really far away, or there's not much dynamics here. But even so, you can see that some of these cells have some trajectories as soon as they're converted, and we're able to capture these trajectories. And we expect that in her case, when she challenges the fish, that there will be a lot more movement and that she will be able to then track their displacement, velocities, and so on. Now, of course, I'm aware that I've given you a good number of examples of what you can do with Mosaic and micropoint, right? So before I sort of end my talk, I'd just like to give you a brief, perhaps a comparison of applications. So a small note to say, where would you choose a Mosaic? Where would you choose a micropoint? Or is there no difference at all? So for a large number of applications, say FRAP or uncaging or photoactivations, both devices are equally good. What is important to remember is that if you're working with multiple regions of interest, then a Mosaic is much better able to do that, because a micropoint is a galvo-guided laser. So it will be able to do these regions of interest, but it will approach that sequentially. Another example would be something like ablation. So if you're looking at doing ablations or axotomies or wanting to make these fine incisions in Drosophila larvae to study development dynamics and so on, a micropoint has the power density to be able to do that, but a Mosaic will simply not be able to deliver that type of power. So in this case, you're much better off with a micropoint. And finally, if you're doing optogenetic activation, type experiments, then it becomes important to remember that the micropoint only covers the central quadrant of your field of view. And this, of course, is in context of it being matched to an Andor front-to-back illuminated SCMOS camera sensors and so on. So there is, a certain element of difference there. But largely, the galvos accurately cover the central quadrant of the field of view. So if you're looking at sort of simultaneous conversion of large fields of view, then a mosaic is much better matched to do that type of application. Of course, in most cases, it doesn't truly make a difference because you can always steer your sample to the central quadrant and then use the micropoint galvos to guide the light to your regions of interest. And with that, I think I'm going to take a breather, say thank you to everyone who's hanging behind to listen to me today. Thank you, Claudia, for inviting me to speak. And just a small sort of shout out to the Insight Awards. So this is a scientific imaging competition that we run every year. And if you use any Andor products, could be cameras, could be microscopes, could be the analysis software, Imaris, which is quite popular. If you use that for your science, do feel free to submit your images to this competition, and you might be able to win up to $1,000. But apart from that, we get to see, all the fascinating science that you're doing in your labs. And as an application scientist, that interests me very much. So I would really encourage you to submit your data sets to this. I've left my email here. Please feel free to take a picture, reach out to me with any questions you have. And if you're going to be at the All Notes meeting in about 2 weeks, then I will see you there in person. Thanks very much.